Hi, I'm Grace Thornton. I'm with the Aquatic Invasive Species Unit with Washington Department of Fish and Wildlife. I'm going to be showing you the environmental DNA unit today. We're going to be running this system through the Columbia River here to get our sample. Okay, so now we're going to take our sample, our environmental DNA sample. We have the filtration system right here. Turn it on, put the filter in the water, and you run about 5 liters of the river water through the system. This takes generally around 10 minutes, and then we will do the negative system after that to make sure there's no contaminations in our machine. In order to avoid contamination with our own DNA or DNA on the dock or whatever, we use sterile gloves. We use bleach wipes, wipe everything down, sterilize it. The filter unit is self-contained, and we'll take that back to the truck after we get our sample and do it in an enclosed environment to try to minimize the amount of external DNA that might be added. The filters then are dried out, and then we send them to the lab at Olympia. We take this environmental DNA, eDNA. We're looking for the DNA of specific species, in this case zebra quagga mussels, but we also are looking for New Zealand mud snails and northern pike. So environmental DNA is free-floating in our water, air, soil of organisms that have shed their DNA. The way that they test this is that they already know the signature of the species that we're looking for. So in our case, zebra quagga mussel. And if that signature shows up in our sample, then you know it's in your system. That indicates that there's DNA present in the system. Doesn't necessarily mean that that organism is here presently, but that their DNA has been in the water system at some point. So the reason we take a negative sample is to make sure that we have no contamination within our system. We run 1 liter of deionized water through the system. So this is the cartridge that holds the filter that we use to collect our sample from the water. We're going to take it up to the truck where it's a little bit of a less windy and variable environment so we can process that. We put it into some desiccant beads, and that goes back to the genetics lab in Olympia. So here's our container for the filter system. We're going to take it apart out here where it's less windy, and there is your sample. You take it out of the filter, fold it in half. I did sterilize everything before I started. And then we put it in these little desiccant beads, which I use the outside of the bag to fold it again, put it snugly in the desiccant beads, and close that up, and that goes back to the lab. And then I do the same thing with the negative, and each of the packets is labeled accordingly. And the negative. And that's our sample. And those two go together in another sterile bag to go back to the lab.